All right, what's up, everybody? Welcome to Omnipotent Health and Fitness episode number 9. Our guest today is a world-renowned pediatric gastroenterologist, research scientist, and entrepreneur. His roles and accolades include being the director of the Center for Celiac Research and Treatment, division chief of pediatric gastroenterology and nutrition, and director of the Mucosal Immunology and Biology Research Center at Massachusetts General Hospital. He is a professor of pediatrics at Harvard Medical School and professor of nutrition at Harvard T.H.Chan School of Public Health. However, with all this success, you may be tempted to think that his life was without difficulty or challenges, and to that, you would be wrong. He started off his research career searching for a vaccine for cholera, but when the vaccine was administered to individuals, it caused the patients to have enough diarrhea to make it clinically unacceptable. After years of hard work, literally and figuratively down the toilet, it would have made anyone want to quit. But like a master scientist, he instead asked what he could learn from this experience. Understanding that serendipity is when preparation meets opportunity, this perceived failure eventually led to the discovery of zonulin and the advancement of our understanding of celiac disease, autoimmunity, and chronic inflammatory diseases. So please help me in welcoming the man whose life's work has helped some of my close friends overcome their celiac disease, and the author of the brand new book, Gut Feelings, The Microbiome and Our Health, Dr. Alessio Fasano. Dr.
Thank you,
Fasano, welcome to the show.
Thank you. Thank you for having me.
Oh, thank you for joining. So I always like to get an origin story from all of our guests. What was it like growing up in Italy, and how did you become this great research scientist and physician?
You know, unfortunately, I'm old enough that, this is definitely a generational gap in terms of the feeling of what was growing up in Italy at that time. That was the boom of, the economics when I grew up in there. But I belong to a very humble family. So, we didn't have too many means. So, but life was great. You enjoyed very little things in life. My father and my mother were extremely busy trying to bring goods to our household, and therefore I was raised by my grandfather. There was a physicist. It is an extremely extravagant individual. He worked in Enrico Fermi's lab. When Enrico Fermi, that won a Nobel Prize for Physics, moved to the United States, he did not move because he was, committed to his family. And then, World War II started, and, Germany wanted him to go there to help them to develop the atomic bomb, and he said, absolutely not. And he ended up in disgrace because he lost his job and all 9 yards. But anyhow, bottom line, he ended up to teach high school physics, and that's my memory when I was a kid. So I grew up with him, and probably that instigated my intellectual curiosity that brought me to become a scientist.
Wow, it's incredible. Just another incredible example of serendipity at its work. So I think a beautiful place to start would actually be what I alluded to in the introduction, and kind of how your research career got started from trying to develop a vaccine for cholera and then eventually the discovery of zonulin.
So again, journey serendipity indeed. At that time, when I was in medical school, I had a mentor that was, instrumental to guide me in this process. And again, silly me, I wanted to really, do something that would be impactful. Since I went to medical school, I knew that I wanted to do research. So I was thinking about something that would save the life of many individuals, work on cancer, that kind of stuff. And then I met this man, and he said, if you really want to make an impact, you have to work on diarrheal diseases. And you got to be kidding me. Diarrheal diseases, this sounds not glamorous at all. And he said, do you have an idea how many kids they die every year because of diarrheal disease? We're talking about 5 million kids, and that was it. So, I studied, tremendously with great details, the physiology of the GI tract. And that's what was my training there. And but I missed the flip of the coin. So in other words, the macroorganisms that make you sick with diarrheal diseases. And he said, if you really want to do this, you got to go to the United States. And you need to go in a place that is called the Center for Vaccine Development in Baltimore. They do a lot of studies on bacteria pathogenesis that deals with diarrheal diseases, and you will learn a great deal of business there. I was supposed to be there for 3 months. I ended up to be there for 2 years, as typically happen in this situation. And one of the things that they put me at work on was to work indeed on a vaccine for cholera. This place was unique in the face that, you know, the approach was to not use what we call kill vaccines, but attenuated live vaccines, so that this will induce the immune response for protection because alive, but will be harmless because you do something to this, you know, microorganisms to not make you sick. Cholera at that time was supposed to give you this purging, very severe diarrhea due to this toxin that's called cholera toxin. So we're talking again at a point in which, you know, restriction enzymes were just discovered and led to the Nobel Prize for an individual from Johns Hopkins in Baltimore. So I used this, you know, biological scissors to cut the gene encoding for cholera toxin in this, you know, Vibrio. And I did everything under the sun to make sure that it was safe. And all the preclinical story seems to suggest that this was a superb vaccine. The animals did not die, they didn't get diarrhea, and so on and so forth. Long story short, it was time for prime time to test this in humans. And we did in volunteers. There were students that for roughly $500 will be locked in a facility, and they will get either a placebo, nothing, my vaccine that I told them, stay reassured, would not make you sick, or you got the real cholera, you got the really sick, but we take care of you. So 2 out of 3 that you got $500 for nothing. And, you know, the outcome is what you said. It was, you know, miserable. And, you know, again, well, the ones they got the real cholera, they got, you know, 10, you know, gallons of diarrhea. So a huge purge. You know, the vaccine that I developed was less than a gallon, but still
right.
acceptable. And, you know, I got drunk for a couple of days. So it was 2 years of failure. But then I started to think, you know, what else is creating, you know, this diarrhea? And I discovered this toxin that, you know, make the intestine leak. And then, you know, studied the mechanism of the toxin I raised. It seems strange that, you know, we have all the machinery to make the intestine leak just to be the target of a toxin. Our reason, you know, probably cholera is mimicking something that we make. And then that's what led in 2000 to discover zonulin, that is this modulator gut permeability, and the rest is history.
Right. It's an incredible story because I really feel like it's sort of a metaphor for life. Every time you hit resistance, you can either have the choice of continuing to get drunk off of, I'm sure, gluten-free wine or something, or you can choose to learn something from the situation. And you obviously chose the latter, which is amazing. And so continuing on with the story, how then does zonulin relate to celiac disease? And maybe for those in the audience, what is celiac disease from a pretty high level?
That's right. So, well, let's start with the latter. Celiac disease is a truly an autoimmune disease. It's like having diabetes, multiple sclerosis, rheumatoid arthritis, and so on and so forth, but it's unique in many aspects. First of all, like any autoimmune disease, it's a recipe with at least 2 ingredients. Now we know there are more. So you have to be genetically predisposed. So you have to have several genes that put you at risk. And then you have to be exposed to something from the environment that is mismanaged by your immune system because of the genetic predisposition. In general, when we're exposed to enemies, what we call non-self antigen, the immune system, you know, attack them and try to get rid of them. People with genetic predisposition to autoimmunity, rather than get rid of the enemy, start to attack their own body. In celiac disease, the attack goes to the GI tract that is destroyed. Therefore, what the gastrointestinal system does for living, i.e. digesting, absorbing foodstuff, doesn't work anymore, and you develop, you know, consequences. Why celiac disease is unique? Because it's the only autoimmune disease for, you know, the trigger. We don't know what makes people sick with diabetes, or, you know, with rheumatoid arthritis, or you know, with multiple sclerosis. It is gluten, this strange protein from grains, that is the culprit. So much so that if you put these people on a strict gluten-free diet for life, because the enemy is not there anymore, the immune system shuts down. We'll remember forever, of course, because there is memory there. So if you're exposed yourself to gluten, everything starts again. But provided you're strictly gluten-free, you do not, experience, the autoimmune attack, because again, the target organ is intestine, rejuvenate itself every week. The stem cells of the intestine are very, very active. And therefore that destruction of the intestine will be healing in relatively short period of time, depending how good they are, in embracing the diet. Then we realized that, genes are important. The environmental trigger important. They are absolutely necessary, not sufficient. We realized that again through celiac disease. The coexistence of these two elements did translate automatically in clinical outcome. We found out, following people over the years, because of risk, let's say family members of people with celiac disease, that they may tolerate gluten for 10, 15, 20 years, and then eventually they break tolerance and they develop, the problem. And that led us to understand that beside these two elements, there are other three elements at least at play. They are highly interconnected. One is the loss of barrier function. So zonulin dependent, that's how zonulin comes into the picture at celiac disease and many chronic inflammatory diseases. So normally these two worlds, the genes that live in our body and these instigators like gluten, they are segregated. They are divided by this barrier, the largest interface being the GI tract. These are large molecules, and under normal circumstances, they would not be able to come through. And the GI tract, like any other tract, any other interface with the environment, is made by single layer cells that in the past we thought they were cemented to each other. So the only way to negotiate the trafficking from the external world into our body was through the cell. Then a Japanese group, like in the early 90s or late 80s, realized that this space is not cemented. In other words, there is no wall in between the cells, but there are doors. Most of the time they're closed, but they can be open and closed for specific reasons that we need. So in other words, to sample the stuff from the environment, we call antigen sampling. But the correct function is to open and then immediately close. So the steady state has to be closed. It's like the door of your house. You want that closed, all the time, but you open it when you need people to come in. But when they come in, you want to keep that closed to stay safe. Zonulin is the key that opens these doors. And, if you produce too much zonulin, these doors, they got stuck open, and then there's this excessive flux of antigens. That leads to engage the immune system, the fourth element necessary to develop chronic inflammatory diseases. So you need hyper-belligerent, immune system. The fifth and last one is the microbiota. So in other words, the composition, the ecosystem in the gut or any other district of your body, but in this case, the gut, will create the condition to epigenetically switch your genetic predisposition to clinical outcome. What that means, that because our microbiome speaks with us through changing our gene expression, you can be genetically predisposed to develop any disease, cancer, autoimmune disease, neuroinflammation, but it's not destiny that you will eventually develop this disease. If you do or do not, depending on how you play your genetic card through the transduction of the microbiome interaction that is influenced by our lifestyle. So you change the microbiome, you epigenetically switch on or off these genes that start the march from genetic predisposition to clinical outcome. But you need to have all these five elements. So you have to have the genetic predisposition, the exposure to something that will make you sick, the leaky gut through the zonulin business, the hyperbelligerent immune system, and the microbiota changes. And finally, parenthetically, interestingly enough, when we ask ourselves, what makes zonulin to be produced in excess and therefore make this intestine too leaky? Two elements. One, gluten. Gluten, some fragment of gluten can do that, but the most powerful way to do that is changing the microbiota composition that will make the production of zonulin to be exaggerated.
Yeah, I've heard you speak about the 5 pillars of developing chronic inflammatory diseases, which were very clearly discussed there, which is incredible. So I guess my follow-up question then is, Is it the ingestion of the gluten that is feeding certain bacterial species, which tend to release more of this zonulin-like peptide that causes more of the opening of the doors? Is that how that works?
No, actually, gluten is a composition of proteins, not a single one, including the gliadins, the glutenins, and so on and so forth. They are weird proteins. You know, if you allow me this parallel, proteins like a pearl necklace, you need to break this necklace in pieces that we call peptide, and then pearls, they are the amino acids, they need to peel it off one by one in order to be absorbed and be used either as an energy source or to be remounted based on our genetic program as our own proteins. We can do that with every single protein that we are exposed to, with the exception of gluten. Why? Because it's extremely rich in glutamine and proline, and we don't have the enzymes to digest completely that. So they are undigested fragments. Two of those have been shown to be capable to instigate zone release from the cells. Now, of course, if you eat gluten, you modify your microbiome. But, you know, that is more, you know, and there are still, you know, questions about that. You know, these are more related to changes that will make you at risk to switch those genes to develop, you know, celiac disease. So, in other words, we have seen following from birth, babies are at risk for celiac disease at some point. Some of these kids that develop celiac disease will lose some protective elements of the microbiome and then acquire an enrichment of hyperbelligerent component of the microbiome, what we call loaded with endotoxins. So in other words, substance that if they make the journey to the leaky gut into, you know, our body, will instigate the immune response that eventually the break intolerance and onset of celiac disease. That's as much as we understand.
Right. I want to touch on a term that you mentioned a couple times because it's something that I feel has a lot of confusion surrounding it, is leaky gut. And I'm wondering, you know, there's a few different camps to this, those who believe that leaky gut isn't real, those who believe that leaky gut is the root of all evils. What is leaky gut, and what, you know, clinically speaking and pathophysiologically speaking?
Now, I don't like the term leaky gut at all.
Yeah.
Because it's been used and abused for those that believe that everything is wrong with us is due to that. And again, you're right. There are polarized camps, and as typically happens in life, the truth is somewhere in the middle. That leaky gut will explain everything is wrong with our health. It's unsustainable and undefendable. But that does not exist, or does not, it's just fake news that also not sustainable. You know, I could understand this attitude until the recent past, when biology was not supporting the concept that, you know, the permeability of the gut can be modulated. You know, I mentioned already this Japanese group that was phenomenally important to really change the entire understanding of the biology of mucosal barriers, and not just, you know, the gut. We're talking about the lung, we talk about the blood-brain barrier. They're all barriers that can be modulated in their permeability. And now we have an understanding how, we understand why, we understand, you know, what are the consequences if this permeability is not totally modulated. So when you have, you know, an excessive increase of permeability in between cells, you have eventually an excess, you know, you lose what we typically do when, you know, remember the parallel that say the door that you want to close and open when you have friends that needs to come in. That's what we need to do when we do quote unquote antigen sampling. So in other words, understanding our surrounding and teach our immune system when to stay put or when to fight. But when you have this opening and people, they come in, even strangers that you don't know, you need to do something about it. And that will create that consequences that can lead on a specific genetic background to a clinical outcome. So I believe that, you know, that skepticism of leaky gut, and again, I don't like the term, I prefer, you know, loss of barrier function, increased gut permeability. It's a concept that now it's recurrent over and over again in biology, in the literature. So I don't think there is any doubt that this is real. The real impact on the pathogenesis of disease, that's still object of discussion. Some people, they believe it's the cause. Some people believe that it's the consequence. But, again, now, and I can give you a couple examples, using animal models, and now clinical trials blocking the zonulin pathway, it seems to suggest that it's an integral part of the pathogenesis and not a consequence of these diseases.
Right. And I definitely want to get into that with liraglutide, but a little bit more into celiac disease before we get there. And I think it kind of ties in very nicely with this discussion of loss of barrier function. And a term that you've mentioned or said that I really like is what happens in the gut doesn't stay in the gut. It's not like Vegas. And the manifestations of celiac disease are actually quite broad. And even I was actually a nurse before I went back to medical school. And even then I was just associating celiac disease with essentially just diarrhea and gastrointestinal symptoms. But really, it's a very wide variety stemming from osteoporosis, iron deficiency anemia, dermatitis herpetiformis. But what I found striking from reading your book was even there might be a link with schizophrenia and autism. And I was curious if you could speak a little bit more about that, because that was really a bit shocking to me.
Yeah, I mean, again, the celiac disease is a classical example of an inflammatory process in a specific district that then becomes systemic. So bottom line, for whatever we understand, is when you have this enemy, gluten coming through a loss of barrier function, and the immune system activates, its weaponry in an inappropriate way. So end up to not get rid of gluten as should be done, and that's happened to everybody else, but start to attack your own body. These heavy weapons, these T cells, they produce chemicals and an immune response that would destroy the intestine. And that's the reason why you have gastrointestinal symptoms. However, for a reason that we still understand not completely, some of these T cells are programmed to leave the battlefield, the intestine, and going anywhere in your body. So it can go to the skin and you develop, dermatitis herpetiformis, can go to your joint and you develop joint pain. And as you mentioned, one of the districts where these T cells, they really, really like to go is the brain. So people with celiac disease may experience headache, may experience, short memory loss, may experience, eventually mood swings, all the way to schizophrenia, autism, and so on and so forth in a very, very, severe cases. How do we know that that's the case? How do we know that this really comes for gluten-activated T cells? Because they have a specific, receptors, specific expression on their surface, or some markers. Specifically, technically they are alpha 4 beta 7 CCR9. That means T cells once upon a time was in intestine, and that's where I was activated. And we have now the capability to ask, activate against what? And the T cells would tell us against gluten. And we found them again in all this district, even in the pancreas for people that develop comorbidity with type 1 diabetes, for example. So I believe this is a remarkable example of how, immunology, molecular biology, epidemiology, genetics can come together in this disciplinary approach and answer fundamental question: why I develop a chronic inflammatory disease, why I develop, celiac disease as an autoimmunity, and what can I do about it?
Right. That it is an incredible example. And considering that celiac disease is sort of this chameleon and a great mimicker of many types of diseases, what do you suggest is the best way to screen for this? So my wife is a nurse practitioner in primary care, and somebody comes in with these vague symptoms. What are the symptoms that she should be looking for to screen with, IgA, anti-TTG, or something like that?
Albert, you just mentioned this. This is a clinical chameleon, so very complicated compared to any other autoimmune disease. I mean, if you pee a lot, you drink a lot, you know already that you have diabetes.
Right.
It's mellitus or insipidus. That's the only question there, but you have diabetes. What comes to celiac disease, I can make an argument that if you pick your nose twice a day, that can be suspicious of celiac disease. I mean, really, it's so complicated. The reality of the story is, your wife, as a wise, nurse practitioner, should have a low threshold to test for celiac disease. That test you mentioned, the T.H.Chan antibodies, cost us $40. And when you have symptoms for which you don't have an explanation, chronic fatigue, joint pain, abdominal pain, and you've done other tests, or even you're going through your differential, one test that you want to throw in there right away is the test for celiac disease. I mean, of course, if you have history, family history, there is somebody else that with celiac disease, even more so. But even in absence of any history, because this is multi-genetic, so you can go back generations and have celiac disease, I would suggest, have a low threshold. And last thing, you start by saying there was a conception that celiac disease was a disease with diarrheal disease, weight loss, and so on and so forth. We rarely see this now. We see more this vague, unknown specific symptom. Why? Because the intestine is a long, long tube. If you have one of the 15, 16 feet of the intestine that is the size of an adult inflamed, chance exists that you're not going to have the classical symptoms because the rest of the intestine can subsidize. And you can even have constipation. You can be obese with celiac disease. If you have, 10 of the 15, feet of the intestine gone, of course, you're going to develop diarrhea, weight loss. But now we see this rarely. In the past, it was much more frequent. But we see rarely that, the damage is so diffuse to express itself clinically with this, purging diarrhea and 20 pounds weight loss.
Right. And to your point, this is a very complicated disease, and to complicate it even further, what is the difference between celiac disease and celiac insensitivity?
So, once upon a time, we thought that if you have problem with gluten, that has to be only celiac disease. And if you rule out celiac disease, you have no business to go on a gluten-free diet. We now know that it's not the case. Celiac disease is the autoimmune response to gluten. Then we have a allergy response to gluten, like wheat allergy. And then the third element to which you alluded to, this non-celiac gluten sensitivity, is still an immune response. It's not, autoimmune, is not an allergic response, but still a response in which you have inflammation without the damage of the intestine. So there is no villi blunting and that kind of stuff. There's just inflammation and give you symptoms totally overlapping, celiac disease. So very difficult to distinguish on a clinical ground.
Right. So, you have celiac disease, you have celiac insensitivity. They can have very similar manifestations and symptoms. Should people avoid gluten, I guess, is kind of the $1 million question. And I know that it can lead to diagnostic pitfalls when you do go evaluate for celiac disease. But beyond just the evaluation, do you feel that gluten is leading to more pathologies than we currently attribute it to?
Many times people, they ask me, because, gluten has this fragment make the intestine to leak, and because gluten has other fragments that induce inflammation, should we all go gluten-free? And the answer is no. The vast majority of people with eating gluten, they tolerate it, no problem whatsoever. And if you and I are on a Zoom meeting now, is thanks to the advent of agriculture, including the advent of domesticated crops like wheat. Otherwise, we would jump like chimpanzees from a tree to another still. So I would not demonize gluten in that sense. Said that, of course, like anything in life, abusing, one element of your diet, so an unbalanced diet, is not good for you. Even stuff that is very good for you, like, turkey meat. If you had too much, you're going to fall asleep, and we know why. So everything needs to be done in moderation, balance, and diversify the diet. That's the bottom line. Of course, if you and you and I don't know if you have any problem with gluten, but if you do not, and neither do I eat an entire pizza. Guess what? I'm going to feel sick. So you will be the same. So everything needs to be done with common sense and moderation. But I would not go that far to demonize gluten as an element that if we don't eliminate from our diet, we will be extinct as a species. I don't think so.
Right. It's a beautiful answer, and something that I'm glad to hear since I love pizza. It's probably one of my favorite foods. But so I know one of the missions that you had early when with celiac disease was one diagnosing it, but another way of going about it was also with your treating it with potentially a pill. How far are we with lorazepam, and what are the early results, or what is it showing?
So the gluten-free diet, we thought that was the most efficacious way to treat celiac disease with no side effects, other than lifestyle, of course. And as a matter of fact, we thought that the efficacy of the gluten-free diet was 100%. Everybody that embraces the gluten-free diet will feel much better. It turns to be not the case. It turns to be that 20-30% they have what we call non-response to celiac disease. They continue to have symptoms despite they are strictly gluten-free, mainly because enforcing the stringency of the gluten-free diet at all time, not just when you travel, when you go to friends, when you go on vacation, is not an easy task. And therefore, the need to find an alternative or complementary way to treat celiac disease has been fueled since, the better understanding of its pathophysiology has been clear to us. Our approach, was if indeed antigen trafficking as one of the 5 pillars is necessary, we stop that and we leave the gluten into the picture. Should we obtain the same results? And the long story short, seems that that's the case. And lorazepam is the zone inhibitor that shut this door for a certain period of time so that you would not be exposed to this excessive amount of gluten coming through. This pill have been having mixed fortunes at the beginning due to the management of the, by the way, you don't do this in academia. Now there are companies that do that. So at the beginning, the companies, they took, some misturn how to handle that. And then, they put the art together. They were, taken over by another company. That COVID came into the picture and slowed down everything. But long story short, it's in phase 3 trial. So the very last phase of the trial. And it is, if this trial will finish, in a successful way, the next step would be commercialization. And I want to mention very briefly, because, we start the discussion with Serendipity. I want to finish with Serendipity. During COVID, of course, we shut down the entire operation as everybody else. And as we did clinically, also research-wise, we repurpose everything. Well, we ask everybody, no matter who you are, you need to focus now your clinical and your research focus on trying to fight COVID. So did we. So I was involved in managing COVID patients from day one, but at the same time, we rearranged the lab to work on COVID. And being a pediatrician, one of the things that we are facing at the beginning was this, what I thought that was a misconception, and turns to be indeed as such, that this COVID, the SARS-CoV-2 was sparing kids. I said, this will be the first time that a virus will spare kids. Kids in general, are the ones that pay, the most of the price when it comes to viral infections. And turns to be that, of course, the virus given infection, but it's almost, with no symptoms. That's the reason why they were under the radar screen, but they turned to be a phenomenal source of spreading of the virus. And now, something that, everybody agreed, but at the point that we made this observation 2 years ago was questionable. But where I'm going with this, that what we learned, that there are few kids that pay a dear price because they develop the worst case scenario in what we call the long COVID. That is this multi-organ inflammatory syndrome, or MIS-C, that it's happening weeks or months after the initial encounter with the virus, with this cytokine storm that really, insult, noble organ like the heart, the kidney, the brain. And we didn't have an understanding what was going on there. And, this is what we learn. Port of entry, airways, that's clear. But after that, in these kids, the virus clears immediately and found in a biological niche where in the GI tract, where it sits there for weeks or months. There causes dysbiosis. Dysbiosis causes zonal release. That leads to a trafficking of some of the SARS-CoV-2 element, particularly the spike protein, the one that we targeted for this messenger RNA vaccine, into the bloodstream. This spike turns to be a super antigen capable to expand the T cells and induce an autoimmune response, because MGHfC is an autoimmune response. So we reason, if that's the case, larazodone may work for this situation, because we had no treatment. We do not have any treatment for MGHfC other than band-aids, steroids, immunoglobulin IV, and so on and so forth. So long story short, we got a approval from the FDA, a compassionate use of larazodone. In one case of MGHfC that went extremely well. This kid was in the ICU. He was able to be extubated and did well. And because of these results, we got the okay for the use of the larazodone in another 4 kids with MGHfC. What was the most striking stuff that we saw, that while to clear the spike protein would take 10 days or so in kids untreated, within 24 hours, spike protein was gone in the one that's treated with larazodone. The results, and clinically they recover much faster. Nobody, end up to be in the ICU and so on and so forth. And the FDA approved a phase 2 trial that is ongoing. Now, this is a story of serendipity, because when, 20 years ago, when, this vaccine failed, somebody would have told me this is what's going to happen as an outcome. I said, you must be out of your mind.
Yeah, absolutely. That's an incredible story. And as a father of 2 myself, it really warms my heart to hear that there is some sort of treatment, and the fact that things can be done so efficiently due to COVID. Essentially, it forced our hands to be much more efficient with how we develop new procedures and treatments. So that's incredible. I want to kind of shift gears a little bit and talk more specifically about the microbiome, and specifically about your new book, Gut Feelings. What was the inspiration behind this book?
Well, mostly Susie Farty and I, she's co-authored this book, were forced in this kind of a venture by the MIT Press. This gentleman that ran the editorial office at MIT said, you are an opinion leader on the microbiome. There's a lot of confusion on the matter. Why you don't write a book on the matter, on the microbiome? And I said, I don't think that this is a good idea. I said, why? Because it's still early. We're still in infancy of the field, and it's stuff changes all the time, and so on and so forth. But he said, there are some fundamental principles that will never change. Long story short, we gave in, and we decided to write this book. It was I believe an experience of love, so to speak, because you really need to be committed to somebody to do this this kind of book. It took almost 2 years to write. Why? Because we finished chapter 1, started chapter 2, and now new papers come out that question what we wrote in chapter 1, and you need to write again chapter 1, and we went on and on and on that way. To put this in perspective, the microbiome literature has been the most prolific literature in human history. We're still seeing the 700-800 papers a day on microbiome published. There's a lot of interest there, and it's an explosion of interest is due to the fact that technology allowed us to study the microbiome as we never got the chance to do before. Now, you mentioned that besides to be a professor of pediatrics, I'm a professor of nutrition, and I grew in a very humble family in the Mediterranean area, where the Mediterranean diet was, pinpointed as a lifestyle that makes you to live longer and healthier. And therefore, I was very intrigued by all this. And I asked to myself, why, the microbiome is so important, and what kind of impingement can be on my health. And when it became extremely clear that no matter what kind of environmental factor you're talking about, preclinical, your mom lifestyle, if she is exposed to pollutant, lives in the urban area or in a rural area, or perinatal. Am I born preterm? Am I born being fed breastfeeding or bottle feeding? Or did I got any infections? Or post-term, my lifestyle, my stress, what kind of environment I grew up in. All this will impinge on the microbiome. So the bottleneck is the microbiome. And the microbiome co-evolved with us since the dawn of our evolution 2 million years ago. The microorganisms were here way before that we were here. And they will be here way after that would be extinct. So we are shaped the way that we are because the consequence of this, negotiation, symbiotic, if everything goes well, relationship with the microbiome that will make us to play our genetic card one way or another, dictating if we live healthy or not, if we live long or not, if we age graciously or not. So that was the impetus. And, I was trying to encapsulate everything in terms of, there is major, xylus, coevolution with the microbiome. What is the consequences if the microbiome is not in agreement with my body, i.e., the disease that you can come up with, if the microbiome is imbalanced, and what is the future in capitalizing on this information in terms of artificial intelligence and mathematical modeling to predict what should I do to really change the microbiota so that I will stay healthy.
Yeah, that's beautiful. And I'm so glad that you were pressured into writing this book. I'm actually waiting for it to come in from Amazon, and I'm very excited to read it. I want to take a slight tangent because I'd be remiss. You mentioned you're a professor in nutrition. We have a world expert in microbiome. I have kind of a few low-hanging questions that I've always been curious about. So I recently started a ketogenic diet. I feel great on it. How does the ketogenic diet affect the gut microbiome? And prior to this, I was doing a lot of intermittent fasting, which again, I feel great on it. Does that affect the microbiome?
Oh, big time. The microbiome is like a farm with different animals. There are cows, there are sheep, there are, horses, there are chicken, pigs, and so on and so forth. They all need to be fed in a different way. So if you change your diet, of course, some animals will be favored because they will have more food to eat. Other will be put at disadvantage that, because they don't have enough food to eat. So what we don't know yet is, what kind of diet will eventually lead to, making the good guys to proliferate and the bad guys to be in disadvantage. And it's very personalized. So I don't think that a ketogenic diet is good for everybody because, or any, vegan or, paleo or whatever. But there will be subgroup of individuals that based on their microbiome composition, their genetic makeup, the many benefits, of that diet. For example, it's well described that the ketogenic diet is very effective to treat epilepsy in people that do not respond to epileptic drugs, and now we know why, because the GABA signaling is favored in a certain way, and so on and so forth. The intermittent diet, I believe, is more physiologically logical because it reflects our evolution. The reason why we have this explosion of chronic inflammatory disease, particularly in people that embrace the Western lifestyle, because this departs tremendously for the plan of evolution. When we were gathered hunters, we were eating not every day. So sometimes you eat, sometimes you don't. Right. You eat mostly stuff that you gather because they're there. They're easy, fruit, vegetable, nuts, tubers, they're in abundance there. So I have just to pick them. Meat, sure, but you have to hunt the animal and you have to find a way to be skilled to do that. So that was more rare. And sometimes for a few hours or a day, you will fast because you can't find either. And that's the way that we evolved. And that's the way that I interpreted the benefit of the intermittent, fasting diet.
Right. One of the missions that you had with celiac disease was a good and non-invasive way to start diagnosing it. Is there something, a test to get an assessment or to diagnose how well our gut microbiome health is currently?
Not yet. There are several companies that are really aiming at this. And again, all this stems with the concept, what is a normal microbiome? We thought that, testing hundreds, thousands, or millions of people, they're healthy, we will find the signature of the healthy microbiome. But as we are all different from each other, even monozygotes twins, epigenetically, they have genes that are expressed in different way. By corollary, by consequence, a symbiotic microbiome is different from each other. What is important is the outcome of this interaction that is fixed for everybody. We all have to have the blood pressure within a certain range, the glucose level in a certain range, and that's these metabolic processes are achieved by this ideal interaction between our gene and the microbiome. When we will have enough data to understand that, and therefore, to interpret the composition and function of our microbiome, then we will be able to achieve exactly what you said. Do the tests that will be non-invasive, that will give them my direction. What I see in the future is the following. You go to your wife's office with a chip with your whole genome sequencing. She put that in the computer so she knows genetically who you are. You also brought, stools that she will analyze in the back of the office. And by the time that she finished the visit you, she will have your microbiome composition and your genome in the computer. And in the deep machine learning algorithm based on modeling that we've been doing for the past 10 years would tell your wife, listen, if you don't change the microbiome, your risk to develop Alzheimer in 20 years is 70%. To bring this risk to 0 or to 10% or 5%, whatever that will be, of course, we can annotate your genes, much easier to annotate your microbiome. This is the kind of diet that you need to go on so that we switch, the composition that way. You go back after the diet, she will check your microbiome, was that goal achieved. Now your risk is down to 5%. That's what I think that is going to happen in the future.
That's a beautiful future that I'm extremely excited for. And we're truly just at the infancy at this whole new frontier that we're going to explore. So this is wonderful. Dr. Fasano, before I ask my final question, where can people follow your work, support your research? What can we do?
Well, again, first of all, this is the results of an extremely dedicated group of individuals. I'm the director of an orchestra, but the maestros are the ones that make all this. So our mucosal immunology biology center, in Boston, and our, European Biomedical Research Institute in Salerno are the two hubs where we do all this kind of stuff. So if you come to our website, we rely on funding from, the NIH, of course, and other government agencies, but a lot of funds that come from philanthropic, donation of people that understand that, if they donate, to our, cause, this will help us to help them and, everybody else that eventually face this kind of problems. And, parenthetically, the two books that you mentioned, Gluten Freedom and gut feelings, the revenues, they all go back to research. So we're not going to make any money out of this. So, and then of course, spread the word. I mean, this is a world that unfortunately see travesties like, wars and pandemics that you will consider the 21st century would be past history, and we never learn enough from history. So I'm the kind of individual who looks at the glass always half full. So live your life, in full, gear in terms of be kind with yourself and the others, eat well, sleep well, and drink good wine, Italian wines. So that would be pretty much the essence of the message here.
I love that. I love that very much. And we'll make sure to put those links in the show notes. So you sort of started to answer it, but my final question that I end all these interviews with is, if you had a magic lamp and a genie came out and you could make the entire world adopt one lifestyle modification that would most impact their health, what would it be?
Nutrition, by far. No question in my mind. Again, I know that I'm biased, but why in the Western Hemisphere we are seeing this, surge of chronic inflammatory diseases, including neuroinflammation, cancer, autoimmune disease, and so on and so forth, because we do not eat the way that we're supposed to. we took 2 million years to build this unbelievable engineered car, this Ferrari that, was built to have in the tank, super octane, gas. And now we put water in there. We wonder why the engine is not performing. So I think that, again, there is no excuse of, eating, stuff that you don't prepare yourself, that you demand to the others to say, well, I don't have the time. If you don't have the time to eat good stuff because you choose and cook yourself, or you don't have the time to exercise, you're not taking good care of your body, and therefore you're going to pay consequences. We have to realign where are the priorities in life. And again, making money, be successful, and so on and so forth, that is, the focus on the Western, lifestyle. It just do not fit the bill, because, all this can be futile, can be, ephemeris, because you're not going to be around anymore. Or if you are, you're going to be on a wheelchair and with tube in your nose and pull-ups because, you poop in your pants. That's not a way to live. You really live a healthy life.
Yeah, absolutely. I love that so much. Dr. Fasano, I want to leave a little bit of time for the Q&A after. So thank you so much for coming on our show. I really appreciate it. And until next time, everybody, thanks so much.